Good morning. It's February 18th and this is your daily brief in science. Here's everything you need to know. A new study shows that rotation inside a fusion device drives edge flows that shape how exhaust particles hit the diverter. Researchers using SOLPS-ITER at PPPL with collaborators from MIT and NC State conducted experiments at the DIII-D facility to test how core rotation interacts with edge drifts. The findings indicate that parallel flow driven by the rotating plasma core influences edge flows at the diverter, a factor needed for predicting exhaust behavior and diverter design in future reactors. When core rotation combines with crossfield drifts, the impact on recycling asymmetries is significantly greater than either factor alone, underscoring the need to include edge flow dynamics driven by rotation in reactor design. The study shows toroidal rotation together with crossfield drifts explains why more plasma lands on the inner diverter target than the outer one, aligning simulations with experimental results. Earlier models that only included crossfield drifts failed to reproduce the observed asymmetry, calling into question the reliability of some design guidance. By incorporating core rotation, the simulations resolved the discrepancy and improved the usefulness of diverter design guidance for predicting exhaust impacts. The work appears in Physical Review Letters under the title Combined Influence of Rotation and Scrape-Off Layer Drifts on Recycling Asymmetries in Tokamak Plasmas, published in late 2025, with funding from the U.S. Department of Energy. ERICEMDEE leads the team, with contributions from PPPL, MIT, and NC State. And the research relies on experimental data from DIII-D and advanced modeling. The results emphasize that predicting exhaust behavior in fusion devices requires integrating core rotation effects into edge flow models to enhance reactor durability and performance. Cellectar Biosciences is expanding its patent footprint to cover ether and alkyl phospholipid compounds for cancer treatment and cancer stem cell imaging, protecting iopofosine I-131 and CLR-125 across Europe, China, Israel, Eurasia, and New Zealand. The move broadens global IP protection for the company's cancer-targeting drug conjugates and enabling technologies, including their radiopharmaceuticals. The new patents strengthen coverage for both therapeutic use and imaging, including dosing regimens for iopofosine I-131 and cancer stem cell imaging, as regulators and investors monitor the path to late-stage development. Context includes EMA guidance, FDA breakthrough therapy designation, and ongoing financing activity shaping strategic progress. The release notes risks such as reliance on future approvals, funding, collaborations, and general drug development uncertainties, and states there are no current product revenues. An FAQ clarifies patent scope, jurisdictions, EMA filing impact, targeted therapeutic areas, and notes the absence of disclosed commercial terms. Background highlights the company's pipeline, including CLR-125 and CLR-225, alongside the broader phospholipid drug conjugate platform. The press release reiterates the phospholipid drug conjugate delivery platform and focus on cancer-targeting treatments with improved efficacy and safety. The strategy emphasizes fortifying exclusivity for iopofosine I-131 and CLR-125 ahead of late-stage regulatory steps, with no financial terms or licenses disclosed. CEO James Caruso underscores momentum and the commitment to delivering new therapeutic options to cancer patients, noting strengthened IP fortification supports long-term commercial plans and patient access goals. The company maintains focus on its PDC platform and pipelines, iopofosine I-131, CLR-125, and CLR-225, with notable designations and trials, including FDA Breakthrough and EMA PRIME for iopofosine I-131. A new approach from Lawrence Berkeley National Laboratory uses artificial intelligence to turn weeks of chemical data into minutes of insight, enabling real-time observation and on-the-fly hypothesis testing during a single experiment. The Digital Twin for Chemical Science, or DTCS, links APXPS measurements with physics-based simulations through forward and inverse loops, powered by computing resources at NERSC and run alongside APXPS instrumentation at the Advanced Light Source. The study, published in Nature Computational Science by Jin Qian and colleagues, uses a water AG 111 case to demonstrate the method. A digital replica of ambient pressure X-ray photoelectron spectroscopy analyzes surface chemistry on operating devices like batteries in real time. In tests, DTCS modeled a silver-water catalytic interface relevant to batteries and catalysis, predicting the appearance of oxygen-containing species on the silver surface within minutes, and matching established results. The platform creates real-time, physics-based simulations that mirror experimental data and forecast future reaction behavior, integrating digital twins with APXPS at the ALS DTCS is framed as a step toward autonomous chemical characterization, with DTCS 2.0 planned to add Raman and infrared spectroscopy and broaden access to researchers at multiple facilities. The project relies on resources from NERSC and ALSA PXPS instrumentation, marking a significant advance in interfacial chemistry insights and autonomous experimentation. The system operates with two linked loops: a forward loop that aligns simulated spectra to experiments, and an inverse loop that infers mechanisms and kinetic parameters for on-the-fly hypothesis testing and planning. DOE Office of Science and Berkeley Lab support underscores a collaborative effort involving ALS, NERSC, and related programs to accelerate discovery through digital twins in chemical science. Built atop APXPS at the Advanced Light Source and leveraging NERSC computing, DTCS rapidly connects theory and experiment. DTCS promises rapid, data-driven feedback during experiments, potentially reducing interpretation cycles and accelerating progress in energy storage, catalysis, and materials science. A new approach to fighting metastatic cancer is taking a major step forward in Rochester. Mayo Clinic and Newphase Limited have installed the first magnetic nanoparticle-mediated hyperthermia system in the United States for cancer research, a heat-based treatment being studied as a potential companion to other therapies. The device is an electromagnetic induction system that surrounds the torso and heats iron oxide-containing nanoparticles delivered intravenously to accumulate in tumors, with a temperature cap of 50 degrees Celsius and cooling to prevent overheating. Installation was completed in November 2025, and the first United States patient was treated in December 2025 as part of a clinical trial for metastatic solid tumors outside the brain. The collaboration aims to expand options for cancers resistant to multiple lines of therapy, with potential improvements if efficacy is shown. This effort follows earlier ultrasound-based heat therapies in the 2000s that faced reliability and comfort issues, prompting a shift to nanoparticle-based hyperthermia. Researchers describe hyperthermia as a potential fourth pillar of cancer treatment, to be used with radiation or to allow lower radiation doses for radiation-resistant tumors. Studies are assessing hyperthermia both alone and in combination with other treatments to slow or halt tumor growth and potentially enable reduced radiation exposure. Lead clinicians Mayo Clinic's Scott Lester and Sean Park emphasize the investigational nature and potential of hyperthermia as an adjunct. The work sits within Mayo Clinic's Radiation Oncology Department in Rochester, with the program noting a financial interest in the technology and that revenues will support its not-for-profit mission. The machine enables treatment of multiple deep-seated metastatic tumors outside the brain, heating tumors selectively while monitoring the patient's body temperature. Overall, the approach builds on historical heat therapy in cancer, using nanoparticle heating to address earlier reliability and safety concerns and reintroduce hyperthermia with greater precision. A Rutgers-led team headed by Daphne Monroe reports a breakthrough in offshore clam farming. Atlantic surf clams can be grown in ocean cages off New Jersey, with spring clams showing faster growth and higher survival than fall. Published in the North American Journal of Aquaculture, the findings suggest offshore farming could boost New Jersey's seafood production while easing pressure on wild clam populations. The cages were designed to shield clams from predators, reduce sediment buildup, and keep them elevated to improve water flow and meat quality, yielding a high-grade product suitable for steam or raw consumption. Researchers note ongoing challenges, including gear durability in rough weather and navigating the regulatory framework needed to scale up offshore farming. The study involves Rutgers scientists Laura Steeves, Sarah Borsetti, and Rachel Davitt, and reflects collaboration with international partners. Funding came from the National Oceanic and Atmospheric Administration. with a partnership involving commercial fishing companies to ensure real-world applicability. Rutgers emphasizes aligning with commercial fishing partners to translate findings into market-ready offshore aquaculture solutions. The results point to offshore aquaculture as productive and environmentally responsible, with potential benefits for local jobs and a more sustainable seafood supply in New Jersey. Hurdles remain before commercial-scale offshore farming can be implemented, including durable gear and regulatory navigation. The study highlights potential economic benefits for New Jersey's farming and fishing sectors as part of a broader sustainable seafood strategy. Offshore aquaculture is framed as a way to meet rising seafood demand, reduce pressure on wild clams, and offer cleaner waters, more space, and fewer conflicts with coastal users than nearshore farms. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.